Hello, my name is Grant Harrison, and I'm April 6th, 2023, and I'm going to present to you the types of sensory receptors in the periphery nervous system and their links to the central nervous system. So there are some simple categories, not to use that term too much, but of sensory receptors in the nervous system. The first being simple, which they are they can be either unmyelinated or myelinated, and these are typically just result in free nerve endings. They're not specialized, and they're typically end up as being nociceptors or sensors for pain in the periphery. And then moving towards more complex, the axons of these sensors are typically myelinated, though not always. And the ends of these sensory receptors, the receptors themselves, are typically enclosed in a connective tissue capsule of some kind, and the shapes of these can change depending on what types of sensor it is. So we do have the ones that have myelinated axons are the Meissner's corpuscles and the Pacinian corpuscles, whereas Merkel cells actually have an unmyelinated axon. The Ruffini endings are myelinated up until the terminal end, but the terminal ends are actually unmyelinated. We also have non-neural receptors. These are neurotransmitters that actually assist in the function of other sensory receptors. They synapse at the sensory receptor itself. Examples of this would be hair cells of the inner ear, and they help to propagate an action potential or help to send the signal back to the central nervous system. We also have accessory structures such as the lens and cornea of the eye, body hair, various structures that help aid in the sensor function. But the text does explain 4 main types, but I'm going to throw in another type, and that's being a nociceptor, because it's not actually grouped in any of the 4 above categories. But anyway, here are the types, and it's mechanoreceptors, chemoreceptors, thermoreceptors, photoreceptors, and then lastly, nociceptors. So mechanoreceptors are probably, I would say, the most common, or prolific. They respond to mechanical forces such as pressure, touch, vibration, and stretch all throughout the periphery, found in layers of the skin, hair follicles of the inner ear, as well as muscle spindles. The communication to the CNS begins via afferent nerve fibers that enter the spinal cord. The primary sensory neuron of these mechanoreceptors either synapses in the medulla, which is more of like the fine touch, vibration, proprioception, or the dorsal root horn of the spinal cord, which is your coarse touch, deeper pressure, and temperature sensations. All mechanoreceptors have a secondary sensory neuron that synapses again in the thalamus before proceeding to the primary sensory cortex of the brain, located in the parietal lobe. And then, so here's a couple of the types of mechanoreceptors. We've talked about already in the class the Pacinian corpuscles. They lie deep in the skin. They detect deep pressure and high frequency vibrations. And then, so you can see the shapes of these actually seem to be representative, and granted they're graphical representations, they seem to be representative of what the type of pressure is. So you can see like the shape of the Pacinian corpuscle looks like it's meant to sense deep pressure. Same with the Meissner's corpuscle. Looks like it's meant to send finer signals, low frequency vibrations. It looks like it's meant to carry that signal throughout that, again, connective tissue nerve ending. Merkel receptors, as we talked about, they're more for detecting pressure and texture. They have myelinated axons, but they have unmyelinated endings, sort of like an eraser on the end of a pencil. And finally, Ruffini endings, they, as you can see, have detect stretch and deformation, and they are going to have, again, unmyelinated nerve endings, but they are still going to be encapsulated in that connective tissue of a nerve fiber. And again, back to free nerve endings, these could be for pain. In this example, the free nerve endings are attached to hair and they sense movement. So another function of a free nerve ending. Oops, there you Yep, there we go. So another type of receptor we have is chemoreceptors, and there's a couple different ways that these function, primarily for smell and taste, at least in the periphery. So we have this aerosolized substance that makes it to the olfactory epithelium. These signals pass along a single dendrite to the cell body and then a single axon to the olfactory bulb via cranial nerve 1. Then that signal is propagated further to the olfactory tract, into the olfactory cortex, and then to the cerebral cortex and the limbic system. Taste buds, similarly, these sensors, they're primarily in the taste bud regions of the tongue. And they contain anywhere from 50 to 100 to 150 taste receptor cells, or TRCs, per taste bud. These TRCs release chemical signals that activate the primary sensory neurons, or the gustatory neurons, and then that signal would travel further through cranial nerves, either 7, 9, or 10 to the medulla, then to the thalamus, then to the gustatory cortex in the deep cortical region called the insula. Another type of chemoreceptor we have is very important, and they're involved in the output of our cardiac output. So we have the glossopharyngeal nerve, the vagus nerve. Those are going to be carrying signals from the carotid body chemoreceptors and the aortic body chemoreceptor back to the medulla, and that's going to help us regulate changes in environmental conditions such as pH, O2, CO2, any change in pressure, or like I said, pH. Those are going to send the afferent sensory neuron to the medulla oblongata to regulate respiration or cardiac output, and hopefully make helpful accommodations. And lastly, almost lastly, we have photoreceptors, which are more numerous. So in photoreceptors, we're talking about the rods and the cones in the eye. So the rods are more numerous than cones. They are just responsible for detecting light and dark, detecting movement and peripheral vision, and they're most sensitive to low levels of light. Cones, they are less numerous than rods, but they provide central vision. They're most sensitive to bright light and responsible for our ability to see colors and fine detail. Our sense of sight is transmitted via the optic nerve through the optic chiasm and synapse in the lateral geniculate body of the thalamus. From this point, the signal is propagated through the neurons that terminate in the occipital lobe, the visual cortex. All the while, a concurrent signal is passed from the thalamus to midbrain, where these synapse with efferent neurons of cranial nerve 3 for pupil dilation. Thermoreceptors, found throughout the body and skin, muscles, internal organs, and the central nervous system. These respond to changes in states of temperature, such as hot and cold compared to baseline. Their sensors exist independently for both hot and cold. It's not just one thermoreceptor that controls both. So that is actually very helpful to us. It helps us be more specific in our regulation of our thermal regulation of our body. And so one of the aspects of these thermoreceptors is that there is a range where there is slow adaptation to changes in temperature, whereas outside of this range, so that's between 20 and 40 degrees Celsius, whereas outside of this range, typically, especially above 45 degrees Celsius, thermal senses may be overlapped with pain. And then that's where we come to nociceptors. This is a response to nauseous stimulus, including tissue injury and inflammation. And then, in essence, these receptors help us generate pain signals. And we have different sizes of nerve fibers that send these signals. And so that also has to do with the myelination level, or the size of the fiber is directly linked to the speed of the conduction. So we have these afferent signals from the nociceptors. They're carried to the CNS via the larger A beta fibers and C fibers being the most common. And then the histamine response or other stimulus may activate a subtype of C fiber. This is perceived as itch. And these are my references. And thank you for watching.